Several recent studies compared the extent to which tomato genotype affects the colonization of plant tissues by Salmonella. Overall, none of the tested tomato varieties were completely resistant to Salmonella. There were 100-fold differences in population sizes of the pathogen on tomatoes and within the fruit tissues. In a limited survey of tomato varieties, there were no detectable patterns in susceptibility of heirloom or commercial varieties/hybrids to Salmonella. However, cherry tomatoes were generally less conducive to proliferation of Salmonella. Tomatoes carrying ripening mutations were significantly less conducive to Salmonella proliferation. The biochemical basis of this phenotype is not yet known. Differences in proliferation of Salmonella in mature and immature tomatoes did not seem to depend on the pigmentation of the ripe fruit. Once contaminated, red ripe tomatoes are significantly more conducive to proliferation of Salmonella than green or partially ripe tomatoes. Cherry tomatoes tend to be less conducive to proliferation of Salmonella compared to larger fruited tomatoes. Generally, susceptibility of tomatoes to Salmonella did not correlate with fruit color or with whether a variety was an heirloom or a modern commercial hybrid. However, levels that Salmonella was able to reach in fruits of different varieties were tomato genotype dependent. Under some field conditions, tomatoes carrying ripening-related mutations were less conducive to proliferation of Salmonella.
The main goal for this research was to test whether different varieties of tomatoes are more or less susceptible to contamination with Salmonella. So we tested a number of existing commercial varieties, heirlooms, and hybrids to see if they respond differently to Salmonella, to learn whether some varieties may be more or less resistant to this human pathogen. And what we found out is that some of the varieties that carry ripening delayed mutations tend to be more resistant to Salmonella. Same thing we found for smaller fruited cherry varieties. They seem to be less conducive to Salmonella proliferation. The long-term goal of this project is to find whether we can use already existing varieties that have reduced susceptibility to human pathogens like Salmonella and incorporate them into the production practices.